really excited to be introducing Trevin and letting him kind of talk a little bit about the work that he's been doing with me over time. So I'll hand the floor over to you.
Wonderful. I'll go ahead and set up my presentation here. All right. Can everybody see that slide all right? Awesome.
Looks great.
Thank you. Thank you so much. All right. So yeah, for the past about 4 semesters, I've been working in the lab with Dr. Brett Schofield, and we have been testing the viability of a yeast insulator assay for screening mammalian proteins. So I want to give a little bit of information about gene regulation because our experimentation ties into gene regulation as a big picture idea. Gene regulation is an essential function of cell survival. Also, all cells and DNA need to carry out this gene regulation because it allows them to adapt to their external environment. The activation or silencing of genes that takes place during gene regulation is essential to produce, the activation of these genes is essential to produce certain proteins that are essential for these cells to survive. There are several different mechanisms of gene regulation, but for now, I wanted to zoom in on heterochromatin and euchromatin being one of those mechanisms. So there are 2 types of chromatin structure. There's the euchromatin that can be seen over here on the left, and the heterochromatin that can be seen over here on the right. The euchromatin is a structure of chromatin that is composed mostly of transcriptionally active genes, and it is more loosely, it's not bound as tightly. Whereas heterochromatin is very tightly packed silenced genes. The majority of the heterochromatin is made up of silenced genes. And what's very interesting about heterochromatin is that it has the ability to spread to adjacent chromatin and silence adjacent genes. So it kind of acts as a wave of silencing. This wave of silencing of heterochromatin that left unchecked would continue to silence this euchromatin, the genes that are transcriptionally active in this euchromatin. So what stops the spread of this heterochromatin? What stops this heterochromatin from reaching this euchromatin? And that is an insulator protein, which is what we're going to be talking about in our research. So why are insulator proteins of interest to us? So the identity of insulator proteins is fairly well known in simple eukaryotic cells such as yeast. However, only one mammalian protein by the name of CTCF has been shown to possess insulator activity. And so these insulators are essential components that allow for gene expression. So they're of great interest to us, especially since we only know of one that has been known to possess insulator activity. So without these, certain essential genes would not have the potential to be activated. Also, there are likely a lot more mammalian insulators than just the CTCF. So we need a way to identify these mammalian insulators and see if they're effective insulators or not. And so we will be looking at a single test in a yeast system to see if it is a viable test to show or identify insulator proteins, mammalian insulator proteins. So now I'm going to talk about the position effect and the spread of this repressive heterochromatin when left alone will spread across the DNA. And keep in mind, this DNA is in a system of yeast. It will spread across the DNA, and in turn, it will silence any genes in its path because of its ability to spread to adjacent chromatin and silence adjacent chromatin. However, when an insulator protein is brought in, it acts as a barrier and blocks the spread of this repressive heterochromatin. And because of this, the gene that is more downstream from the insulator protein that it's protecting is able to be activated. And what this means is this gene is now able to produce the proteins that it is needed to survive in certain environments. So if the strategy that we're going to be looking at is to be successful, mammalian proteins must retain their insulator activity in yeast. And we are going to test this by looking at CTCF orthologs. So if you remember, CTCF is that only mammalian protein that has been shown to possess insulator activities. And so we are looking at that same gene, but from different species. And we are going to actually be placing these candidates, these orthologs that we're going to be testing in between these two genes so we can monitor when their genes are activated, and we can look at growth patterns of that gene and determine whether or not the insulator was effective at insulating. And so as we can see by this image, our positive control is going to be perform robust growth, and our negative control will perform minimal growth. I'd also like to point out that each column, as you move from left to right, has 10 times fewer cells in it, and that's kind of why it seems to die off. We wanted to get different, at least 5 different dilutions. And so with the positive control, we will see growth, because if the insulator was effective, it is able to stop that spread of the heterochromatin, and the activated gene can produce the proteins needed to survive on this media that we used. And the negative control will show minimal growth, and we can determine that an insulator protein was not there because that heterochromatin was allowed to travel down and silence the genes adjacent to it. So here are our results. These are the 4 orthologs of CTCF that we used during this assay. And we actually used CTCF from a chicken, a mutated CTCF from a chicken, or sorry, a human, mutated CTCF from a human, a fruit fly, and then also a chicken. And you can see the negative control and positive controls acted as shown in our last slide. However, none of the orthologs that we tested showed insulator activity. And this was actually unexpected and kind of peculiar. We obtained unexpected results by this because we were expecting at least one of those orthologs to show insulator activity, because CTCF is our only positive control. We would expect that to show robust growth and thus show insulator activity, because the positive and negative controls grew as they were supposed to. But the fact of the matter is, none of the orthologs showed insulator activity. So where do we go from this? We ask the question, is CTCF actually being expressed or made in these experiments? Because if it's not, obviously that cell is not going to have that insulating barrier. So that's what we're doing now. In the lab right now, we're performing some ongoing research. We are currently conducting an immunofluorescence procedure to the yeast cells. And by doing this, we use fluorescent antibodies that we can then attach to what we're looking for with fluorescent tags. And we're able to look at these under a special kind of microscope and detect where these things are that we have tagged. So we'll be able to visualize whether or not this CTCF was actually being made throughout our experimentation. And here's an image of a immunofluorescence unrelated to our experiment, but just to show you, the scientists here tagged elements that were to appear in the cytoplasm out here with a red immunofluorescence tag, while the elements that were expected to be seen within the nucleus were tagged with a green immunofluorescence tag. So that's just kind of showing how we will look at these images. So what we got from this experiment is, until we receive the results from this immunofluorescence procedure that's ongoing, these results suggest that this insulator assay may not be an effective way to screen mammalian proteins for insulator activity, given none of our expected insulator candidates showed that insulator activity. It also can tell us that mammalian insulators may not retain their function in yeast. This system may not be a viable system to test for these mammalian insulators. But we are concerned, as I said, we are currently continuing our research using this immunofluorescence to detect if CTCF is present or not. And this research will find us an answer that allows us to determine if there has been this missing piece of our research in our previous testing without the CTCF being expressed. And until this immunofluorescence procedure is completed, we won't know that. But so far, these results seem to indicate that the yeast assay may not be an effective way to screen mammalian insulator proteins. So with that, I'll go ahead and open it up to any questions that anyone may have. And so thank you so much for listening, for your time.
Fantastic presentation. Thank you. So if there are any questions, please go ahead and raise your virtual hands. Perhaps while people are thinking, I can ask about why pick CTCF from different species? Why not just use one?
I suppose we were trying to get a large array of variables that we could test. We would want to test different proteins from different species, because if those orthologs were to show insulator activity, who knows what other species of orthologs can show insulator activity. So not only are we finding these in mammals, but perhaps orthologs of those genes could be found in other animals as well. So I think it was just testing as many species as four different species to see if we could detect insulator activity in all four of those.
Fantastic. So let me just give people a moment. If you have questions, go ahead and raise your hand, or frankly, there's so few of us, go ahead and unmute yourself and ask.
I will unmute myself and ask. So I have kind of an opposite question. So why have you chosen yeast as the, I guess, what was the word you used? I guess as the system to test this.
Sure.
Are there other options besides yeast?
Sure, absolutely. There are other options besides yeast. The reason that we are working with yeast is because it is so easily to genetically manipulate. And so we were actually able to create plasmids that connected these candidate proteins to the DNA by placing them in between these genes. And this was a lot easier to do in yeast than it would have been in, say, mammalian cells or higher level eukaryotes. But yeast is just so easy to genetically manipulate, and that was the main driving reason for us using this system of yeast.
Makes perfect sense. Thank you.
Yep.
All right, fantastic. Let's all give our speaker a virtual round of applause.